This video is about protein structure. Protein structure is important because the shape of a protein dictates its function. If you need a review on what proteins are, check out our video on proteins and amino acids. The way amino acids are arranged in a polypeptide chain is what determines the shape of a protein. As a little side note, RNA is what tells the amino acids to bond in a specific order, and this occurs on the ribosomes of a cell. Protein structures are broken down into 4 levels of complexity: primary, secondary, tertiary, and quaternary structure. All proteins have a primary structure, but there are many proteins that do not have a quaternary structure because they simply aren't that complex enough. The primary structure goes as far as the sequence of amino acids in a polypeptide chain that makes up the protein. The secondary structure tells a little bit more about the shape. Let's go over 2 common types of secondary structures, the alpha helix and the beta pleated sheet. The alpha helix is where the polypeptide is twisted into a helical shape, and this is supported by hydrogen bonds in the amino acids between a hydrogen from the amine group and an oxygen from the carboxyl group. Next, the beta pleated sheet is where the polypeptide chain has folded in a way that 2 chains are next to each other. Hydrogen bonding takes place between those chains, and so the space between those chains is kind of like a sheet. The tertiary structure describes the 3-dimensional folding that occurs in the proteins and what kind of interactions between the R groups influence that. Let's look at 4 common interactions that occur between R groups as part of the tertiary structure. First, we have hydrophobic interactions. Remember, hydrophobic means to repel from water. Hydrophobic interactions occur when nonpolar R groups clump together inside a molecule, which hinders that molecule's ability to make contact with water. Now, when these R groups are clumping together, van der Waals interactions occur between the R groups that further influence molecular properties. Some examples of van der Waals interactions are dipole-dipole, dipole-induced dipole, hydrogen bonding, and London dispersion forces. Next, we have ionic bonding that can occur between the charged R groups. Last but not least, the tertiary structure can tell us about disulfide bridges. These are very strong covalent bonds that occur between 2 cysteine molecules. Cysteine is one of the 20 amino acids. Cysteine contains a thiol group that has the ability to form a disulfide bond if it's next to another thiol group. The 4th and last level of complexity is the quaternary structure. In the tertiary structure, we were looking at an interaction between R groups. Here, we are looking at interactions between polypeptides. Quaternary structure describes the subunits of a protein, if a protein is that complex. For example, the quaternary structure of hemoglobin shows that it has 4 polypeptide subunits. That's all for this video. Thank you for watching and good luck studying!